So, hello everyone, am I audible? Okay, so let's start. My slides are visible to all of you. Am I audible and my slides are visible?
Yes, ma'am.
So, what we have studied yesterday, we have studied about this overview of gut microbiome composition. We have studied that there are so many phylum or there are so many phyla which are in abundant quantity in the microbiome, and it is influenced by so many factors like age, diet, location, geography, and the antibiotic, whether we are taking any kind of pharmaceutical products or not. So all these things will influence the microbiome, normal microbiome, and it is known as the dysbiosis. When it is altered through different environmental factor, it is known as the dysbiosis. So we will study a little bit more detail about what are the major phyla which are available in the our body. So the major phylum are firmicutes. If you talk about the abundance level, the major is firmicutes. Apart from that, bacteroidetes, actinobacteria, proteobacteria, and verrucomicrobia. So firmicutes are gram positive bacteria and bacteroidetes are gram negative bacteria. So first of all, you should know about the different microbial diversity in a body. normal composition of microbiome. So you can see these two are the major composition, like firmicutes are about 64% and bacteroidetes are about 23%. So firmicutes are what? They are firmicutes are gram-positive bacteria, and majorly they include firmicutes include Bacillus species, Clostridium, Lactobacillus. These are the major genus under this firmicutes. Now 23% is about the bacteroidetes. So bacteroidetes are gram negative. So you have to remember that this firmicute is gram positive and this bacteroidetes are gram negative. So basic comparison you should know about it, that bacteroidetes are gram negative and these are gram positive. Now these ratio, that is the firmicutes about bacteroidete ratio, is of great importance for a medical perspective. Your doctor will check your F by B ratio if he or she wants to know that whether there is a disturbance in the normal microflora of your body or not. So all these things, like an increased F by B ratio, an increased F by B ratio means if your firmicutes and bacteroidetes ratio is very much increased, then it may be associated with obesity and different gut diseases like irritated bowel syndrome or inflammatory bowel diseases. So they will check about this F by B ratio. Now these are different examples of this firmicutes. You can see here Lectospiraceae, Rhinococcaceae, and Lactobacilli, Enterococci, firmicutes. So all these are majorly identified by 16S rRNA gene sequencing. So just because of the help of bioinformatics and the 16S rRNA sequencing, we are able to know that our microbiome is composed of different microbial diversity, not only just single bacteria, but so many different bacteria. You will study and it is very, very interesting. So see, in contrast to limited phylum level diversity, the diversity of genera and species in the mammalian gut is enormous. So there are different kind of species and different kind of genus you will see in a single gut of a mammal. Like you can imagine that as per the 16S RNA gene sequencing, the data is about 1800 genera, 16,000 species, and more than 36,000 strains of a normal intestinal diversity. So it is very, very huge in number. Like, can you imagine that you have more than 1800 genus, only genus we are talking about. And if you talk about this species, it is more than 16,000. So it means 1800 genus, you can't even imagine. This is so huge. So a single mammalian gut in different kind of microbes. If you talk about in numbers, you can say that more than 1800 genera, more than 16,000 species, and you can say more than 36,000 strains are there. Now genus is different, species is different, strains are different. Like if you say, I think you should know about Bacillus subtilis, a simple bacteria, a simple gram-positive bacteria. Now Bacillus is the genus and subtilis is the species. And many a times you have read that Bacillus subtilis subspecies is also mentioned many a times. So this subspecies is unique for this strain. So this is known as the first strain, and second strain will be Bacillus subtilis, and some other number like KC310 or ATCC, MTCC. So the little variation in their whole chromosome makes it unique. So that is known as the strain. So you can imagine 36,000 strains, more than 36,000 strains are there in a microbiome. Now, in a simple words, what we have studied till now, that human beings have 10 times more microorganism in their 10 times more microorganism in their body. 10 times whatever cells they have, suppose they have 1000 cells, for example. So microorganism will be 10K. Microorganism will be 10K. So this is clear. Now, out of those 10K microorganism or 10x time microorganism, there may be 36,000 different strains. So Archaea represents by a phylotype closely related to methanogen, yeast, fungi, protist, make up only a minor part of the community. Like major are these two, firmicutes and bacteroidetes. Your doctor will also check your F/B ratio. Now see, you can write this part. Somewhat surprisingly, mammalian gut communities are composed of only a few phylum and show a species composition distinct from that of any free-living microbial communities. Suppose I can explain this point in a very simple language, that suppose from an environment we have air, water, soil. These three resources are there in the environment majorly: air, water, soil. Suppose in the air 100 different type of microorganisms are there. Suppose in water you have isolated more than 200 microorganisms, and in soil you have isolated 300 microorganisms. These numbers are just for an example purpose. So these numbers are just for an example purpose. What I am trying to explain all of you is that our body gut, that mammalian gut, composed of different kind of microorganism, which is not at all related to whatever we have in our environment. Like microorganism in our body is not at all equal to what we have in the environment. It is totally dissimilar from whatever we are having in the environment, whatever strains we are having in the environment. So why this is? Because microorganisms are coexisting in the living body, and here in the environment all these things are abiotic factors. So accordingly microorganisms are different. But when we talk about an human body, which is a biotic factor, so the microorganism will be different. So the majority, 98% of all human gut phylotypes fall into one of the four bacterial groups. Like firmicutes, 64% of your total microbiome, bacteroidetes are 23%, proteobacteria are 8%, and actinobacteria are 3%. So first rank will be given to firmicute, second rank will be given to this one, proteobacteria will be third, and last rank will be given to the actinobacteria. Arrange it in a decreasing order or an increasing order. Increasing order: actinobacteria, proteobacteria, bacteroidetes, and firmicutes. And if it is asked in decreasing order, firmicutes will be first. So these four we have to study. Now, there is a high variability from person to person in microbial species abundance of the gut community. We have read this yesterday also, that a person A will have some different microbial diversity with the person B. Why it is so? Because of the different food habit they have, what kind of environment they are producing in their body. It depends on the kind of lifestyle we are having. Obviously, the lifestyle of one person varies from the other one in terms of the food habit, in terms of geographic location, in terms of physical activities, and also if we are taking any kind of pharmaceutical drug or not. So, studies of a healthy men and women, like in between 27 to 94 years of age, have revealed that each person harbor but few hundred to no more than 1,000 of many thousands of different human gut microbial species detected, and that a person's species composition is relatively stable over long period. So this paragraph is nothing complex, don't worry. It means that out of the four phyla I mentioned in the previous slide, a study suggests that if you compare men and women microbiome in the age group 27 to 94, will have all these four phylum, thousands of each of the phylum. It means suppose thousands of firmicutes and 600 of bacteroidetes, 200 of actinobacteria, and likewise. So these are the major bacteria. It is still under exploration, but majorly these four are abundant in different microbiome of a men or in women. And comparative studies have also shown that humans share more genera with each other than with other species of mammals. So I have mentioned this point yesterday. Whatever I mention, I always include the bullet point that person, human being will be matched with human being only. Human being will not be matched with cow, goat, animal, or any other genus, because genus should be same. Then only your genome will match with the person. Like our genome will be matched with our parents, but not with our cousin, or our grandparents, or immediate paternal and maternal things are shows similarity in between the sibling and the parent. So likewise, human 1 will have similarity with its microbiome to the human 2, but not with the monkey, cow, or any other animal. So this suggests that precise mammalian gut microflora is fine-tuned to each mammalian species. Now, the major contribution of gut microorganism to human metabolism. This is the function. What is the role of the microorganism in our gut? What they do there? They are huge in number. There are more than 36,000 genera with 36,000 strains we have studied just now. So what they do there in our gut? So human gut microorganisms synthesize a large variety of enzymes, which is a bioproduct that allow for the processing of complex dietary carbohydrate into monosaccharide. So as you all know, we eat polysaccharide. We eat polysaccharide like rice, chapati. So a cell, if you talk about a human cell, so if you want energy, your cell is unable to decode that polysaccharide. It has only capacity to digest and synthesize and communicate with monomers. It can only sense monomers, that is glucose. So this is a cell. You are feeding it with polysaccharide. So what it will do, it has a machinery, it has a metabolic machinery that it can convert the complex polysaccharide into simple monosaccharide. So this is not achieved by de novo. They do this with the help of different machinery, by the help of enzymes. So one common enzyme is alpha amylase. Alpha amylase, which is responsible for the hydrolysis of alpha 1,4 glycosidic linkage, which is present in starch and other glycogen and other related polysaccharide. So alpha amylase will cleave this alpha 1 4 bond in the polysaccharide, and then it will release what? Glucose, dextrose, and maltose likewise. So the key point is that our body needs monomers. Our body needs monomers to do the daily energy expenditure. So they readily converts the polysaccharide into monosaccharide, that is glucose. So for that reason, we need enzyme. So enzyme will be synthesized by whom? Who will synthesize the enzyme for this function? These microorganisms will synthesize the enzyme, because microorganisms have a brilliant capacity to synthesize enzyme by using organic substrate. So substrate, how they get the substrate? Through the food we are consuming. We are consuming organic food like chapati, roti, rice, whatever you are feeding your body. Basically, if we talk about the balanced diet, carbs you eat, protein you eat, fat you eat, and vitamins and minerals. So all these things are utilized as a substrate. Substrate you are providing, fuel you are providing for the microorganism. They use it as a substrate and they convert it into bioproduct. That bioproduct is known as enzyme. So this feature is utilized by the industry. Now I am one of them. I use microorganism. Basically, my PhD is in enzymology, industrial biotech. So I use the microorganism to synthesize enzymes, but I do it extracellularly. But in our body, it is done through this substrate. They convert it under different condition into enzyme. So these enzyme will be used for the conversion of this complex dietary carbohydrate into monosaccharide and the production of volatile fatty acid, that is VFAs. So I think this point will be clear for all of you. Now, the strain common in human adults have many genes whose product help to catabolize polysaccharide, consistent with these bacteria being adapted to gut environment rich in polysaccharide. So now tell me that nature has everything in a planned manner. Nothing is random in nature. We just see, we click pictures some randomly, but nature has planned everything in a very, very, very fine manner. So suppose if a bacteria, suppose there are 5 bacteria, bacteria B1, bacteria B2, bacteria B3, B4, and B5. So only 3 have the capacity, B1, B2, B3, to hydrolyze the carbs, carb into monomer. When we talk about monomer, ultimate product is the glucose. Our body can only sense glucose. So now tell me, this B1, B2, B3 will be more or less in the gut. It will be higher or lower in the gut. Of course, it will be higher, because in the gut we need bacteria that is capable of converting the complex polysaccharide into monosaccharide, so that energy expenditure will be met for our body. So the bacteria offering this function will be in higher amount in the gut. So bacteroid strains common for the conversion of polysaccharide into monosaccharide will be in higher amount in the gut. So this is the foremost function of microorganism to boost our human metabolism. Suppose if there is a person, how to draw a person? Suppose this is a person and he or she is suffering from any kind of IBD, inflammatory bowel disease. So if you check the enzyme levels or the critical tools which are required for the metabolic degradation of the polysaccharide or whatever polymer you are having, so it will be in very low amount. That is why this person is not able to properly digest the food, and therefore its energy will be low. So this is the principle behind this. We need microorganism to digest our food, to release energy from the complex polymer we are ingesting. So microorganism perform this function. So gut microorganism also function in nitrogen metabolism. So out of the 20 amino acids we require, 10 are said to be essential nutrient because we cannot synthesize them in adequate amount. Although we obtain essential amino acids such as lysine from food, these nutrients may also be produced and excreted by certain gut microorganisms. As we all know, there are some biomolecules which our body is not able to synthesize in an adequate amount. So for that also we need some microorganism, some specific microorganism who are capable of synthesizing that particular requirement. So the solo role of microorganism is to ease our or to comfort our or to support our de novo metabolic machinery to digest each and every polymer we are ingesting. Now, gut microorganisms are also known to contribute to the maturing of the gastrointestinal tract. This includes triggering the expression of genes involved in nutrient uptake and metabolism in gut epithelial cell, priming the immune system early in life to recognize the gut microflora as non-foreign, and the development of mucosal barrier to colonization by foreign bacteria. So these lines are a little bit lengthy, but it is very, very important. First, in this slide, you have read that how a microorganism is our digestion process to convert the complex polymers into monomers. Now, in this, our gut microorganisms, which you can term it as a hero, what it do? Why they are regarded as hero? Because they contribute for our well-being. They contribute for the healthy life of an individual. They what they do, they mature the gastrointestinal tract by triggering the expression of genes. The expression of genes is stimulated by these microorganisms to increase the uptake, nutrient uptake. Nutrient uptake will increase, then what it will happen? It will meet the energy expenditure. Now, apart from that, it also guides the immune system that don't consider us as a non-foreign. Just as you all know about immunity, our immunity recognizes between self and non-self. If you have read the immunology book, that is the QB, you have studied this line that our immunity is able to differentiate between what is self, what is non-self. They can differentiate क्या अपना है क्या अपना नहीं है, what is ours and what is not ours. So on the basis of that, our immune power works. So if these microorganisms are considered as non-self, our immunity will trigger it. Our immunity will be triggered and our immunity will start degrading them. So our gut microorganisms say that don't harm us. We are your friend. We are helping your gastrointestinal tract to convert the polysaccharide or the polymer into monomer, and we are helping you in every possible manner. So that hint is given by the microorganism to our immune system that don't consider as non-self, we are non-foreign. Because if our immune system will consider those microorganisms of the microbiome as non-self or non-foreign or foreign, so they will trigger the response towards it, they will eradicate them. So it is very much necessary to consider them as non-foreign. Non-foreign means self. It is not at all harmful for our body. So all these three functions are performed by the gut microorganism. So see how beneficial they are for us. But what we are doing, we are harming them. We are consuming unhealthy items on a regular basis, and we are destroying our helpers. We are destroying our heroes. So studies of experimental colonization of germ-free mice with individual microbial species or microbial communities have demonstrated that colonization triggers the expression of genes for glucose uptake and lipid absorption and transport in the ileum. Now, all these things are in research, everything is found out by having some sort of experiments. Nothing can be concluded without any sure shot experiments. Like if I want to say that this particular drug is effective against a particular type of disease, so I can't say uh, like without any experiment confidently. So for that, we need to have some sort of experiments. We need some sort of experiment. So all these things which are revealed in this line are only because of certain experiments published in different literatures. So through that, it is evident that the colonization triggers. Whose colonization? The microorganism colonization. If there are so many microorganisms in our gut, they will trigger the expression of genes for glucose uptake and the lipid absorption and transport in the ileum. So this indicates that there may be a link between gut microbial composition and the ability of the host to harvest energy from its diet. So there is a direct correlation between gut microbial composition and the ability of the host to harvest energy from its diet. So all it depends on food to energy conversion. Food act as a fuel. Food act as a fuel. So if your body is capable of converting this food into energy efficiently, then it might be due to the gut microbial composition. If a person whose microbiome is disturbed, you can see that person will have always in a lacking of energy. So this is the condition with obese person or the person having obesity, and we can focus on this now. So an interesting part we can see role of gut microorganism in obesity. So up till now we have studied about what? We have studied about that different composition of quick revision. We have studied that bacteroidetes and firmicutes predominates the whole microbiome, and these are the four groups, four phyla, which are mostly available, mostly found in different microbiome, whether it is a microbiome of a man or a woman, doesn't matter, but these four are there. And also we have studied the microbial diversity, that 1800 genera, 16,000 species, and more than 36,000 strains are there. Now we have studied about the contribution, or you can say the function of gut microorganism. So what they do, they help us to synthesize, to digest the food and to metabolize the food to convert it into monomers and ultimately the energy. Now we have studied this, the other function of the microorganism, the three major function. Now we will study an interesting part, that if a person is obese, what will happen to it and what is the difference between a normal individual and a diseased individual. So for that, I have prepared one cute diagram to make you understand easily. So this person is obviously obese and this person is lean and thin. So everything will be unfavorable for this person. This person will always be unhappy and this person will always be happy. I'm talking in only in terms of gut microbiome, not overall happiness, sadness. No, I'm only talking in terms of gut microorganism and the microbiome. So there are two persons, one is obese and one is thin. So what will happen? So whatever I have written in this slide, I have made this diagram to explain you in a very simple way. So the role of gut microorganism in obesity is to do what? So suppose there is an experiment in which they found that the researchers found that if there is no gut microflora, if this gut microflora is absent, lower body fat will be there, and this has been done in an mice. Its gut microflora has been removed, and then it was grown for a few weeks. Then it was found that its body fat is low. So how this is? It means that somehow the gut microbiota is playing a role in the obese person in increasing the weight of that particular mice. On the other hand, we always have controls in the experiment. Always, if you are a researcher or if you do any kind of research, you know that our every experiment is supported by a control. If you have treatment group, you can't say with surety if you have not run the controls with it: positive control, negative control, no template control, no organism control. So everything we compare it with the control. So you can see here the gut microflora present. If in the normal mice they have not done anything to this mice, so its gut microflora is present, and they have seen that higher body weight. So they have concluded. What will be the conclusion? Simple, that gut microorganism is playing a role in obesity. So they are doing what? They are changing the F by B ratio, and they are changing the amount of enzyme we are producing in our body. Suppose slow metabolism. You have heard about this term, slow metabolism. So why this is so? This is because of the microorganism, inactive microorganism, or the abundance of such microorganism who are capable of holding or absorbing lipids. You know about lipids. Lipids are ultimately, in a simple language, fat. So if those microorganisms are high in number who are able to hold the or store the fat, then our cell will become what? Fat. So this obese person has that microorganism in its microbiome who are capable of doing this thing. But that microorganism is absent in this lean and thin person. So therefore, his weight is low. Suppose if his weight is 80 kg and its weight is 45 to, let's say, 50 kg. So why this is so? Because his cells are healthy. They have not stored fat globule in their cell. So cells are healthy. They are doing every metabolic functions easily, but his cells are having stored fat globules. So they are not able to do the normal metabolic machinery. So in short, the gut microflora diversity plays a major role in the obesity or increasing the weight of that person or not. So I hope this point is clear. Now let's get back to this slide. What I wanted to convey, I explained it through the diagram so that you can understand it in a very easy manner. Now, role of gut microorganism in obesity. So obviously obesity is a significant health risk, because if obesity is increased, cardiovascular disease will increase, and other complications also, diabetes will increase. And if diabetes will increase, diabetes is not at all harmful as per my perspective. The secondary complication associated with diabetes, like the kidney disease, the different pathies, neuropathy, the nephropathy, and different kind of pathies associated with diabetes is harmful. So if you are an obese individual, you will have an always the high frequency of different kind of diseases like high blood pressure, cardiovascular diseases, and diabetes. I have written it here. So gut microorganism may play a part in human obesity, although mechanism remain unknown. It is still yet not revealed fully, but there are so many instances available in the literature indicating different kind of different kind of methods that plays a role in the increasing the obesity of an individual. So gut microorganism to host fat accumulation came from studies using germ-free mice. This experiment, I explained the things which are available related to the role of gut microorganism in obesity are only because of this experiment, germ-free mice and the normal mice experiment. So in these experiments, normal mice had 40% more total body fat than those raised under germ-free condition. If they are germ-free, their body fat is low, and if they have the normal microbiome, their body fat is 40% more than the total body fat of this one. So after germ-free mice were inoculated with fecal material from a normal mouse, they developed a gut microflora and their total body fat increased, although there had no been changes in the food intake or energy expenditure. Mice that are genetically obese have microbial gut communities that differ from those of normal mice with 50% fewer bacteroidetes, a proportional increase in firmicutes. Now see, if they concluded that if it is germ-free mice, lower body fat. So they also conducted one supplementary experiment that if they insert the fecal material will be rich in what? Microorganism, gut microorganism. So they have inserted these microorganisms into this rat, and after a few weeks they saw that this is also developed. Sorry, they have inserted it in this. So what they saw, now it will have increased weight. So ultimately, the gut microbiome or the gut microorganism is playing a role to increase the body weight of that mice. So you can see role of gut microorganism in obesity. So this is lean mice and this is of obese mice. So what you can see here, we can see here that bacteroidetes are red in color, firmicutes are blue in color, and methanogens are green in color. So the differences in gut microbial communities between lean and obese mice, lean and thin mice and obese mice, the comparative study of the microbiome composition of lean mice and the obese mice. So I can draw like this. This is the smiley and this is the obese one. So you can see the microbial composition. What you can see here, obese mice have more methanogens, a 50% reduction in bacteroidetes, and a proportional phylum-wide increase in firmicutes. Nutrient production from fermentation is higher in obese mice due to removal of H2 by methanogens. Now see, whenever you eat, many a times we say we have eaten rice. That's why I feel sleepy. Why it is so? This is very important because whenever we eat glucose-rich food or sugary food or carbs-rich food in a very high quantity, always in our body, we will have fermentation. That is why whenever we eat sugary food or starchy food, we feel sleepy, we feel lethargic. Why? Because our body microbiome or the gut microorganism who can perform fermentation like the methanogens will do the fermentation, and that is why we will feel sleepy, because they will release what? They will release the fermented byproducts. So in the obese mice, they have more methanogens, a 50% reduction in bacteroidetes. You can see these red one are the bacteroidetes. So in the lean mice, it is high, and in the obese mice, it is low. And the food is fermented and hydrogen is released, and then volatile fatty acid and nutrients are made, and in this they are producing methane because the methanogens are high in number in the obese. So this is the reason that there is a difference in the microbial composition of an lean mice and an obese mice. And obese mice will have higher amount of methanogenic bacteria, and it will do fermentation. So it will feel lethargic, no energy, nothing to do. So this is the major difference. I have included one more diagram. Yes. So role of gut microorganism in obesity is, I have explained it. Obese individual had lower proportion of bacteroidetes and a higher proportion of actinobacteria, that is from phylum Firmicutes. You can see here a 50% reduction in bacteroidetes. Obese mice have a 50% reduction in bacteroidetes. Bacteroidetes are this red one. You can see there are so many red ones. If you count 1, 2, 3, 4, 5, 6, 7, roughly we can count 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and about 25 approx. And in this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Approx 16 or 15. So bacteroidetes are decreasing in the obese, and also Firmicutes are increasing. So Firmicutes by bacteroidate ratio is changing. This part is very much important. I will ask MCQ tomorrow related to this, so pay attention. Mouse models, obesity was associated with phylum level differences in the gut communities and a lower bacterial diversity. So you can see the study also indicate that inheritance of the microbiome from mother was a more significant factor in obesity than host genotype. Obviously, the genetic factor plays a major role in each and every biological thing. So, although the exact role of the gut microflora in obesity remains unknown, it is likely that the microbial gut community governs the ability of the host to harvest organic nutrients from its diet. So, you can see this picture. I have made this for in comparison or to let you know about the factors influencing obesity through the gut microbiome. So overall, you can also compare this. So the gut microbial community, low bacterial diversity, phylum level differences, family obesity pattern, microbiome transmission, maternal, all these things play a role in the gut microbial community. And also one major factor is host genotype, and under the host genotype genetic influence will be there, individual variation will be there, human 1 will be different from the human 2, and the lifestyle choices, what we are feeding our body, in what condition we are living, and dietary habits. It is also a part of the lifestyle. Environmental factors like pollution, everything contributes to the different composition of the gut microbiome. So you have to consider each and every factor while considering the microbial diversity of an individual. You can't just say that these are the 4 factors which will contribute to the microbial diversity of an individual. No, there is a long story. So there are so many factors, like genetic factor will also play a role, and obesity, obviously if you are an obese individual, the gut microbiome composition will be something different. And these are the major factors: the lifestyle choices, the dietary habits, and the environmental factors. All these 3 factors are the major, or you can say the regulators of the microbial diversity in a microbiome. That whether this bacteria will be there in your microbiome or not, it will be decided by your these 3 things: whether you are having a healthy lifestyle or not, whether you are eating some healthy foods or not, and what kind of environment you are living. If you are living in a polluted environment, you will definitely have a very imbalanced state of the microbiome, because microorganism too requires a healthy condition to live. Healthy condition means a well balanced state of pH, temperature, and macromolecules and the biomolecules. So everything should be in a controlled manner to offer the growth of the gut microbiome. So now you can see here the contribution of gut microorganism to human metabolism is basically in the production of short chain fatty acids, the enrichment of specific lipopolysaccharide LPS, and the production of vitamins and essential amino acid. So these three are the key points. Now, if an individual is obese, so this SCFA will be increased in this person. It means the microorganism responsible for the production of the SCFA, that is the short chain fatty acids, will be in higher amount in the obese individual than compared to the lean individual. So this point should be taken into consideration while answering about the role of or the core function of a healthy gut microbiota of an individual. So they also, I have also mentioned this earlier, the biodegradation of polysaccharide also, or you can say any polymer to monomer, and the enrichment of specific lipopolysaccharides, the production of vitamins and essential amino acid, and also to boost the immunity. This is also a role of the microorganism, to consider the microorganism as what? Non-foreign. Don't write foreign. Foreign are antigens, which are externally present, and they invade our immune system when they invade the physical barrier, like when they penetrate into our inner skin, that is dermis. So our microbiome consider or give signals to the immunity that don't consider as a foreign material or a non-self material. We are for helping the different kind of metabolic machinery. So this is also done by the gut microbiota. So all these are the function of gut microbiota. Now this is again the same thing, that genetic factors, environmental factors all play a role in the obesity of an individual. This was taken from a paper, a research paper was there. So see, for making a notes or for anything, you should always have a look on what are the different kind of papers available. This is not for UG students, I can say, but for PG student, it is must. You should have an habit of checking that what is going on in the research field, because in every book, even if you are preferring the for another book, it is more than enough. It is very good. But if you want to know that what are the experimental prospects of a topic, so for that reason you should refer some literature, at least a review paper, to get an insight that what people are doing related to this topic, because this will clear your concept, and also many times it will be interesting if you will have an habit of reading research paper, review paper, so you will always have a plus score in your result, because through reading literature we come to know about the different basic things or the conceptual topics of our subject through the paper. And also they have so many different diagrams, like this is a beautiful diagram to explain the dysbiosis of gut microbiota of obese individual. So see, the genetic factors, the environmental factors, the energy absorption, central appetite, fat storage, all these factors play a different role in the obese individual. Like the fat storage will be increased in this obese individual. They regulate the lipopolysaccharide layer of that obese individual, and also the inflammatory genes will be stimulated, and the circadian rhythm also will be influenced. So everything you should study from both book and paper if possible. So this is all for today. I hope this is enough for the day. Now let me open the syllabus. So we have studied this overview of gut microbiome composition and function. Now we will take one more class tomorrow to brush up all these concepts in a very confident manner. We will solve MCQ related to this. Then we will be done with this module 1 because this is an introduction. But still, I tried my best to offer you so much of this unit. So accordingly, we can go up to one step higher in the after this. So tomorrow we will take this unit and after that we will jump onto the second one. So this is all for the day. So let me stop the recording.